I'm Danny Winder. I'm the Bixler-Johnson-Mayes Endowed Chair in the Department of Molecular Physiology and Biophysics and in Psychiatry and Behavioral Sciences and Pharmacology. And I'm the founding director of the Vanderbilt Center for Addiction Research. Our research focuses on the interactions between stress and reward. We're very interested in the problem of addiction and interested in the role that stress plays in that problem and plays in two ways. One is we think about the role of stress in relapse. Maintaining an abstinent phase is the biggest barrier in treating the problem of addiction. We can generate abstinence in addicts, but we can't maintain it. And so we try to think about ways in which stress facilitates the end of that abstinence phase, the relapse. And that can happen in two ways. One is a stressor in the person's life, losing a job, domestic issues, something like that can precipitate relapse. And then the other is an interoceptive stressor. So the withdrawal from long-term exposure to a drug can cause the individual to have feelings of anxiety, depression, things of this nature that can facilitate drug-seeking behavior. And so we're really concerned with the underlying mechanisms that are involved in those behaviors. So to accomplish that sort of research, the tools that we use are tools aimed at understanding specific circuits in the brain involved in addiction-related behaviors. And so much of our work focuses around the use of animal models, such as rodents in particular, where we use electrophysiological and fluorescent recording approaches to be able to track the activity of individual neurons, either in an ex vivo slice preparation or in vivo while an animal behaves. And that is taking advantage of a new wave of technologies that has come from the BRAIN Initiative. I've always been interested in long-term changes in behavior, in learning and memory. So how is it that we can remember things that we experienced decades ago in our life? And so neuroscience in the 90s and 2000s was particularly concerned with the hippocampus and its role in learning and memory. And increasingly, neuroscience has realized that the forms of plasticity in brain circuits that are involved in classical forms of learning and memory also are likely to be involved in pathological forms of learning and memory like addiction. So in addiction, we learn to associate drug use with specific cues and contexts and re-exposure to drug paraphernalia. Basic science is key to our ability to perform addiction research. The technologies that we can now employ to answer questions as to the specific molecules that are involved in addiction-related behaviors are really techniques that did not exist even 5-10 years ago. So the rapidly evolving field of technology development in the basic sciences has been key as we explore new neural circuits and their involvement in addiction-related behavior, and then will also be key as we move to take the next step to, having identified these targets, now move forward to try to develop new ligands to try to treat the problem of addiction. Funding is critical to our ability to perform the studies that we're interested in working on to try to find new therapeutic avenues for the treatment of addiction. We are funded primarily through the National Institutes of Health, but funding from all sources is really vital to our ability to have an impact on this major problem in our society. We are now in a position in neuroscience where we can essentially ask and answer about any question that we can think of in the field, and really the limit is money. So the ability to afford to do these experiments, because the technologies and resources that we need to do these things are quite expensive. But we're in an amazing time in neuroscience right now with the very new technologies that are available. So in the last 10 years or so, there's been a revolution in neuroscience in terms of the ability to specifically image different populations of neurons, either in the intact living nervous system or in fixed tissue in ways that we couldn't do before. So we can now obtain, for example, 3D images, fluorescent images of an entire animal's brain, of a mouse brain or a rat brain, to in fine detail resolve the pathways that have been activated during some aspect of that animal's experience. There has been the availability of optogenetic and chemogenetic strategies brought forth, so that we now, essentially using light or specific molecules, have ways of remote controlling very specific populations of neurons within the brain. And again, because of the utilization of genetic technologies and stereotactic surgeries, we can then deliver those control vehicles into very precise parts of the brain to very specific neuronal populations within those areas, so that the specificity of the questions that we can now ask that are answerable is amazing compared to what it was just a decade ago. We've been really excited by some of the new models that we've been working on in the lab. We work on a model of chronic alcohol ingestion in female mice, where they are allowed access to alcohol in their home cage for a long period of time. This is a study where they're not forced to drink the alcohol. They have a water bottle and an alcohol bottle. And what we were fascinated by was that after a 6-week period of exposure like this, if we then replaced the alcohol bottle with a water bottle, we saw the emergence of an affective phenotype in these animals. And so what I mean is that by a variety of different simple behavioral tests, from what we could see, the animals exhibited higher levels of anxiety and depression, or these are depression-like behaviors. We have to infer this because we can't ask the animal how they feel. And with that comes the ability to then perturb that system and ask how can we intervene in this process, and ultimately ask whether that then has efficacy in treating alcoholism. And so we've been excited that a drug such as ketamine, we found, was able to reverse these affective phenotypes in our abstinent animals. And ketamine is a very interesting drug. It's a drug that can be abused and is an anesthetic at high concentrations, but at low concentrations can serve as a antidepressant, particularly in treatment-resistant populations. And so we're excited about the possibilities there with that drug, although it's complicated. And then another drug is a drug that targets the endocannabinoid system in our brain, and we found that drug to have some efficacy in treating these symptoms in these mice as well. And so we're excited to now be able to work through the neural circuits that are involved in mediating those behaviors. So tech addiction is something that has been in the news a lot lately. There are many types of behaviors that individuals can sort of develop a compulsive seeking towards. And so gambling is another notable example, and our phones are ever present in our lives. And this we may actually be able to model aspects of this in rodents. A former postdoc of mine, Chris Olson, several years ago developed a novel model in rodents called operant sensation seeking. And this was a task where he trained animals, or rodents, mice, to lever press for random patterns of flashing lights. And essentially the mice would lever press for these lights to essentially the same degree that they would lever press for cocaine infusions. And so one could manipulate that operant sensation seeking in much the same way you could manipulate the behavior related to a drug of abuse. And so we think that there are common mechanisms in the brain, common reinforcement mechanisms for behaviors that drugs of abuse can tap into, but that compulsive aspects of our behavior that don't necessarily require ingestion of a substance might also be able to interact with. I was attracted to Vanderbilt when I was considering places to set up my lab by the incredible reputation Vanderbilt had for basic science research and the opportunity and interest in moving that basic science research towards the clinic. And was also attracted to Vanderbilt by the highly collegial nature of all of the faculty here. So this is a fantastic place to do science because it's a lot of fun to work with colleagues whose lab doors are open and who are interested in working together towards common goals. I'm really excited about mentorship and training graduate students and postdocs, and think that this is probably one of the very top goals that we can have as faculty at a basic science institution. They need to be excited by the opportunity to do an experiment where they will know a new fact that no one else in the world knows for that period of time before they've written their paper describing those results. And they need to make sure that they are chasing their passion, and I encourage them to stick it out, because this job is hard, but I think all jobs are hard, and to develop mastery and success requires just sticking it out and taking our lumps as we go along and keep sticking with it. I did my undergraduate work at North Georgia College. It was actually a commuter school, and in between my junior and senior years of undergraduate, I got interested in science. I thought I was going to be a veterinarian first, but then I had a summer research experience at a lab at Emory University that got me really excited about the nervous system, and did my PhD in neuroscience at Emory University and focused on mechanisms of signal transduction in the central nervous system, and grew my interests in synaptic plasticity. And there I was encouraged to think broadly about where to do postdoctoral research, and took that advice to heart and applied all over the country, and was fortunate to get the opportunity to do my postdoc at Columbia University with Eric Kandel, and learned a lot about the novel approaches to studying synaptic plasticity in his lab and how to apply that to the analysis of animal behavior.